Alright, ladies and gentlemen, this is going to be our next video on membranes and specifically going over diffusion. Let's get started on this next video. So if we're thinking about what diffusion is, let's go through and look at these four molecules here. Now these molecules start compacted together, and if I give them enough time, they're going to spread themselves out in this box. For the simple answer of diffusion, this is what diffusion is. It's going from a high concentration of molecules to a lower concentration. They're naturally going to want to spread themselves out as far apart as possible. So diffusion is the net passive movement of particles, atoms, ions, or molecules from a region in which they are in a higher concentration to regions of lower concentration. This requires no energy, and this naturally goes through and happens. Now let's go through and take our box and do a different scenario. We're going to take the same four molecules here, and we'll go through and we'll put and divide the box with a line where the molecules cannot go through and pass through. What's going to happen? Well, they're going to go through and they're going to spread themselves out based on the size of the box that they are given. And if I get rid of this, well, they're going to spread out even further, like they were before. So the molecules are going to diffuse based on the size and the area they are in. Again, this is not requiring energy. This is naturally going through and happening. Now, our last example here is we're going to provide a dividing line here, and we're going to have 5 molecules on the left. And in this dividing line, it's big enough for the molecules to pass through now. Now, what we have here are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 on the right. What do we think is going to happen? Well, the molecules are going to naturally want to go through and spread themselves out as equidistant as possible. So molecules will diffuse until an equal amount is achieved on both sides. This is the process of diffusion. And we can see we have 9 and 9 molecules here on the left. And the reason that is that's going to go through and allow them to be equidistantly spread apart. A word that we give this is what we call equilibrium. Equilibrium has been achieved. And in the next graphic, we're going to take a look at this so we can understand it more. All right, so let's go through and look at this graphic here. If I go through and I put more molecules outside of the cell, and I let them go through and naturally diffuse, what do we think is going to happen? Well, they're going to move and they're going to want to spread themselves out as equally as possible, so they're going to move over to the side with less molecules. So once they achieve an equal amount of molecules on each side, we have achieved equilibrium. Let's do the opposite scenario here. So I put more molecules inside the cell, and I see what happens when they go through and diffuse. Where are they going to move? They're going to move outside of the cell, so that way they can go through and spread themselves out as far apart as possible and be equidistant. So by looking at this graphic, we can see diffusion in process dependent upon where the concentration gradient is, and we'll discuss that on the next set here. So now that we understand the concept of diffusion, let's understand what selective permeability is. Now, selective permeability is a property in which cellular membranes only allow certain molecules to enter or exit. If you're selective about the type of food that you eat, you're going to be picky about it. Same thing for the membrane. They want to control what goes in and out of the cell. Now, things like water molecules, we look at the membrane. Some molecules are going to be small enough where they can go through and be selective for, and the membrane can go through and allow them to move through. It's going to be based on polarity and size. If they're small enough, they're going to go through and they're going to move through the membrane. So in this case, these blue molecules here, they're small enough and they contain the right polarity, so they're going to go through and move through the membrane pretty easily. The membrane's going to go through and keep out larger molecules that it doesn't necessarily want in or wants to regulate. So we can see this molecule is too large to fit through, and it may not have the right polarity, so it's not going to be able to enter into the cell. However, let's say I want to get some of these yellow molecules into the cell. Well, I can use something called a transport protein. The proteins that we've gone through and looked at before. I can open that gate and I can select for the specific molecules that I want inside the cell. So selective permeability is just the selection of certain molecules. It's going to be regulated by the phospholipid bilayer and by the proteins, the transport proteins in the cell membrane. All right, so let's go through and look at how cells are going to transport stuff in. What we see here and what we've been looking at is what we call a concentration gradient. We have an area of high quantities of molecules and an area of low quantities of molecules. Now, if the molecules are small enough, they're going to, again, want to spread themselves out evenly. So they're going to naturally go through and move inside the cell. So we can see these blue molecules here are going to go through and they're going to diffuse into the cell until they achieve equilibrium. So this is just simple diffusion when the molecules go through and move via the phospholipid bilayer. Now, if we look at this, we want to get these yellow molecules in here, but again, they're too big to pass through the membrane. We're going to have a transport protein, which is again going to use the process of diffusion, and it's going to allow these molecules to pass through easily when the gates open, and we call this facilitated transport. So how are cells going through and transporting? Well, what we're talking about now is there's two concepts we need to understand: diffusion through the membrane and facilitated transport via proteins. Now you're probably asking, well, what happens if the concentration gradient, or let's say we have an unequal amount of molecules on the inside of the cell versus the outside of the cell? So like we discussed before, if we want the molecules to move from point A to point B, we can use the natural process. We can move with what we call the concentration gradient. We can move the molecules down. They're going to naturally diffuse and no energy is going to be needed. In this scenario on the right, we want these four molecules to move from point A to point B. This is going against the concentration gradient, and naturally the molecules are going to want to move this way. We don't want that to happen in our cell. We want to gather as many of those blue molecules as possible. When this happens and the molecules naturally want to diffuse to A, we're going to use energy. No longer are we going to use the process of diffusion and naturally let them flow in. We're going to use energy and special pumps and special proteins that will use a concept that we'll look at in a later video called active transport. Okay, so that's a lot of information there, and we want to make sure that we understand how cells go through and move things in. So let's go through and look at this flow chart to try to kind of put this all together. There's two concepts that we learned about. The first concept being diffusion. Diffusion is just the natural net passive movement of molecules, particles, whatever it is, from an area of higher concentration to an area of lower concentration, and it does not require energy. Cells want this to happen so that way they can move stuff in the cell and limit the amount of energy that they are using. So they're going to use diffusion and this natural process and this physical process to move stuff across the membrane. And this concentration gradient is an important vocabulary word for us to know because they're moving with the concentration gradient to get stuff into the cell. Now, the other concept that we learned about in this video is selective permeability. Selective permeability is the regulation of things entering into the cell. So if the molecules are small enough, they're going to go through and move without a protein. They're just going to move through the cell membrane. And the cell membrane is going to regulate that diffusive process there. And they're naturally going to move via diffusion. If they are too big, they are going to need a protein, a transport protein. And now we give this a special word. When they diffuse through a protein, we call that facilitated transport. Without the protein, we call it diffusion. With the protein, we call it facilitated transport. All right, so there was a lot of concepts in that video in a very short period of time, but these are the things we want to make sure that we understand. We want to make sure we understand what diffusion is, how cells are going to use diffusion, and then we want to understand concentration gradient, facilitated transport, all of the vocabulary associated with this video. So what we're going to do next is we're going to go through and look at a video that has to do with active transport when the cell has to get things into it going against the concentration gradient. This is going to be the end of the video. I will see you all in class tomorrow.